Okay, good morning all. Welcome back to the talk 9 of our Astrobiology Webinar series. Our today's speaker is Dr. Ravikumar. He is from University of Agricultural Sciences, Dharwad. Dr. Ravikumar is an entomologist. He has spent around 8 years as a postdoc in NASA Ames Fruit Fly Laboratory. So he has a lot of interesting things about what the fruit flies does in microgravity. He was also involved in half a dozen space flight studies of NASA trying to get clues for understanding astronaut health using Drosophila models. So we have more signs of flies to listen today. I'll put it on to Dr. Ravikumar now. Dr. Ravikumar, over to you.
Yeah. Thank you, Dr. Bala, and I should also thank Dr. Shrija for giving this opportunity to talk about what I have done at NASA Ames Center for close to 7 years. I spent there using these fruit flies as a model system for understanding the astronauts' health in space. Of course, this is kind of a little bit deviating from what you have been listening to the Astrobiology Webinar series. It's kind of space biology work. But there's a lot of crosstalk between space biology and astrobiology. But space biology mainly deals with understanding the impact of microgravity and other space environmental factors on already well-established organisms on Earth, right? So, and like as I said, there's a crosstalk between space biology as well as astrobiology. It's interesting to know the another facet of biology, that is space biology. So we all know that since the evolution of Earth, since 4.5 to 5 billion years ago, everything changed. Like carbon dioxide level, oxygen level, relative humidity, temperature keep fluctuating. The only factor that remains throughout the constant throughout the evolution of Earth is gravity. So that means every living organism on Earth is evolutionarily adapted to the Earth's gravitational force. As a result of that, so it will have a very high dependency on the gravitational force of Earth. So if you remove that gravity factor from the scene, you will have a devastating impact on various levels, be it on cell level, molecule level, tissue level, organ level, or whole organisms level. So the question is, how do you create this microgravity in order to understand the impact of microgravity and space flight environment on the organisms, right? Of course, you'll not be able to do that anywhere on Earth because everywhere you have this gravitational pull, it will prevent you from the microgravity environment. And of course, there are also some simulators like rotating wall vessel and other stuff. You can use it for understanding the simulated effect of microgravity on Earth. But however, if you want to study the real microgravity and real microgravity impact on organism or cells, on tissues or organs, you need to go away from the space. You need to go away from the Earth and try to understand the impact of microgravity on living organisms. So luckily, we have this, the microgravity platform called International Space Station, which is a national laboratory of US, and this is nothing but basically an artificial satellite which revolves around the Earth at an altitude of 400 kilometers, and since 2000, at least 3 and maximum 6 astronauts have been staying in this International Space Station because it is pretty big. Actually, it is as big as 2 football ground size, and many of our space biology experiments will be done here, including our fruit fly experiments we carried out when I was working at NASA Ames Center. So this is very fantastic facility for doing the real microgravity impacts on various organisms, especially biological models as well as various physics experiments and chemistry experiments and material science experiments. So next obvious question is, why do you want to study this microgravity and space environment in general, right? You might be aware that there's a historic landing of Curiosity Mars rover on the Martian surface. In fact, I was very lucky to be witnessed when I was working at NASA Ames Center, the landing of this Curiosity on the Martian surface. And more recently, a couple of days back, another Mars rover, Perseverance, have been launched by NASA, and hopefully it will have a smooth landing on the Martian surface and give us much needed information on the habitability of that planet. The reason why many of these space agencies as well as the private industries like Jeff Bezos and Elon Musk and various other people were interested in spending too much money on the space exploration mission is they wanted to have, or they wanted to make this Homo sapiens or humans as a multiplanetary species, right? So that is the ultimate goal. So hopefully, in the next century or 2 centuries, we're going to colonize possibly Mars. Our best bet is Mars. So if you want to colonize any planets in the solar system, especially Mars, you need to understand a comprehensive understanding of biology in terms of what is the impact of space environment on various levels, right? Cell, tissues, and organs and organisms levels. So that's where these model organs play a very important role. But before we colonize any of the solar system or any of the planets in the solar system, we need to comprehensively understand the health effects, right? Health effects of spaceflight and microgravity environment. We know that from the International Space Station studies, there's plenty of literature available. You can always go through that. There's a huge impact of space environment on human health. I can quote you a few of them here. For example, if you take the cosmic radiation, right? This cosmic radiation can directly penetrate your cells and damage your cell macromolecules such as proteins, lipids, DNA, and especially if they damage DNA, that can cause some kind of mutations that will lead to the cancer kind of diseases, which is really not a good thing to be experienced, right? So that is one issue. Another issue is bone and muscle loss in the space or in general International Space Station or in general space station in space condition, right? So according to the NASA's data, if you are staying on the International Space Station, you're probably going to lose 1 to 2% of your bone mass and 20-25% of muscle mass, which is huge. But the good news is these are reversible and also preventable. That's why you can see astronauts spending at least 2 to 2.5 hours physical exercise on the International Space Station so that they can keep this bone as well as muscle health intact on the International Space Station or space in general. Then we have another issue, cardiovascular issues, right? You can see that heart doesn't have to pump against the gravity on the space, right? On the International Space Station or in the space. So as a result of that, there is a lot of information that indicates there's an impaired function or cardiac diastolic and systolic movement is highly impaired in space, and that will result into the loss of some of the cardiovascular muscles as well as structure, which is again well reported in various animal models as well as in the astronauts. And another issue we have is compromised immune response. In the sense, so basically we know that the astronauts came back from both International Space Station as well as Apollo missions. They were highly compromised immune response in terms of fighting against the fungal diseases, bacterial diseases, viral diseases once they came back to Earth. That clearly indicates the astronauts which are exposed to the space environment were having compromised immune function that will lead to the various kinds of diseases once they come back to Earth. So I'm going to talk about more on the immune response as well as cardiovascular issues because we did quite a bit of studies using model organism on these two topics. In the next few slides, I'm going to talk about more detail on these topics. So another big issues, and most recently we have been seeing in some of the astronauts, is macular degeneration. So basically what will happen is when you are in the space, the body fluid will flush to your head region. As a result of that, then that will put a lot of pressure on the back of your eyeball. That will lead to the intracranial pressure, increase in the intracranial pressure. That will lead to the finally either partial blindness and complete blindness. These are some of the very fundamental health issues we need to be addressing before we even think of colonizing any planets in the solar system. And apart from these, you also have a psychological stress. For example, astronauts are confined in a very isolated area in spacecraft for the longest period of their life. As a result of that, it has a lot of psychological stress. In addition to that, this physical damage will definitely cause huge problem of health issues. So we need to be addressing these issues before we think of colonizing any planets in the solar system. So that's where these model organisms play a very important role, right? So we cannot play with the astronauts' health, as we know that, because their life is very precious. We need to be very cautious about that. A lot of ethical issues. All we can do is we can collect their blood samples, urine samples, and fecal samples, and do some superficial science that may or may not be helpful, because if you look at the data, so far only 559 people have gone to the space. And among them, only 8 have completed 300 days missions. which is statistically very, very negligible. We cannot depend on this kind of skewed data to extrapolate to the entire population which are planning to go to the space in the near future, either through the space tourism or any other mechanisms, right? So that's why these microorganisms and C. elegans, fruit flies, mice, model organisms are very, very critical in understanding the cellular and molecular mechanisms of fundamental issues of health caused by the spaceflight and the microgravity environment. The reason why you want to use these different model organisms, if you want to provide a strongest argument in favor of particular hypothesis, you need to have as many as diverse model organisms data so that you can easily and confidently and very reliably extrapolate to the human health. And that will give you a lot of information about the in general human health issues, right? So when I worked at NASA Ames Research Center between 2011 to 2018, in our lab, we mainly use these fruit flies as a model system to understand how exactly we can exploit this very tiny model organism for understanding the astronaut's health indirectly. So that I'm going to talk about more detail. The next question obviously is how comparable in terms of biology with the humans and the fruit flies, right? If you look at the central nervous system, respiratory system, systemic response, digestive system, excretory system, cellular response, every damn function is conserved in fruit flies as well. In terms of genetics, there is significant overlap between those two. Also, for example, there is a website called Homophila, which is nothing but basically Homo sapiens and Drosophila. They combine to call it Homophila. In this website, you can see that the individual genes of the Homo sapiens have been compared with the Drosophila melanogaster and come to the conclusion that around 60% of the genes have highly conserved in Drosophila as well. So specifically, if you look at the genes which are causing diseases in the humans, have 77% similarity. That is an amazing fact. So which clearly shows that the Drosophila model is very valuable and very reliable model to understand the human diseases in specific, and in specifically human diseases, because there's a lot of overlap between genes between the humans and Drosophila melanogaster. And another advantage is low requirement for volume and crew time, right? The spaceflight always have a space is the biggest constraint in spaceflight. You need to be having a smallest package of payload that can give you the best science, right? So and Drosophila experiments fits into that field. So that is a very important advantage we have using the Drosophila model. And of course, the multi-generation studies can be done. We used to do 1 month experiments typically on the International Space Station. Like within 1 month, we used to get 3 generation flies. The reason being, these flies have a lifespan of, although lifespan is around 40 to 50 days, but life cycle is only 10 days. From egg to egg, it will take only 10 days. That means within a month, you can get 3 multi-generation studies can be easily done, which is impossible with any other higher animal models like mice or rodent models, right? So that is another advantage, because these kind of studies will help us to understand adaptation as well as evolutionary mechanisms by doing the multi-generation studies. So another biggest advantage of fruit flies for the spaceflight studies is statistical reliability. We used to send around 15-20 flies in each wire, and we used to get back around 100 flies. So that is really big number. That will give a lot of statistical reliability compared to animal models like rodents, where you are sending 1, 2, 3, 4, 5 maximum, right? So statistical reliability is very important in case of reliable understanding of that particular phenomena or research. So these are some of the unique advantages we have in terms of using fruit flies for spaceflight studies. And of course, since Thomas Hunt Morgan used these tiny fruit flies for his genetic studies in 1906 until 2020, 10 researchers have received 6 Nobel Prize in physiology and medicine. That is kind of that for their contribution to the human biology in general, directly or indirectly. That will kind of sums up why and how relevant this fruit fly model for understanding not only fundamental biology but also the human diseases specifically. So with that background, I kind of try to convince that how relevant this fruit fly model for in general biological studies and human biology. So if you look at the history of animals in space, various kinds of animals have been flown to the space since 1950s, right, up until 2020. Microbes, C. elegans, fruit flies, mice, monkeys, chimpanzees, then plants, various invertebrates, all kinds of animals have been sent to the space. Laika, the dog, that is a very famous, a celebrity doll, right? Which was spent on the Sputnik in 1957. But however, interesting fact is exactly before the Laika became the celebrity dog who went to the space, exactly 10 years before that, in 1947, February 20th, fruit flies were the first animal to went into the space. This is again, there's an interesting story here. They used this V2 rocket, which is an abandoned rocket by the German Nazis during the World War II. They refurbished that, and through that rocket, they sent to the space using these fruit flies, right? So technically, they went to the space for about 108 kilometers and recovered live flies back by using the parachutes. So according to the definition of space by NASA, if you are going beyond 50 miles, which is roughly around 100 kilometers, then you will be called as astronaut or space, or you are in space. So flies were the first animals technically to went into that benchmark, 100-kilometer benchmark, and flies were the first animals. So since then, up until 2000, sporadic fruit fly experiments have been carried out on various missions, either through the European Space Agencies as NASA, or various other agencies have tried these fruit flies from 1950 to 2000. Very sporadic experiments have been carried out. But the focus at that time was many of them were trying to understand fundamental biology aspects, like how space flight or microgravity or radiation impact mutation rate, recombination, genetic kind of studies, and some of the phenotypes they observe, like mortality, how it impacts the death of these flies, and the fecundity, that is nothing but laying capacity, egg laying capacity, developmental aspects, aging, behavior. Not many of them have even thought of using these fruit flies for understanding more detail on the human biology and specifically astronauts' health point of view. So after 2000, since the era of International Space Station, many of these researchers started focusing on their work using fruit flies for understanding the human health and specifically astronauts' health, more specifically astronauts' health, right? So among them, Dr. Shamla Bhattacharya's group, and she has contributed immensely to this area of research by sending series of fruit fly experiments to the International Space Station, such as FIT, heart flies, FX, and fruit fly lab of series of experiments. So I'm going to talk about each one of these in the next few slides in my presentation. However, I was very lucky to be part of this team actually during that 2011 to 2018, and many of these experiments were carried out during that period. And it was my fortunate. I think I was very fortunate to be part of this team, and with Shamla Bhattacharya working there at NASA Ames, that gave me a lot of experience doing spaceflight experiments, especially to the International Space Station. So the very first experiment Dr. Shamla Bhattacharya's group did was FIT, which is nothing but fungal pathogenesis, immunity, and chemogenesis. This particular experiment was carried out on Space Shuttle Discovery, STS-121. In fact, an interesting fact here is that Sunita Williams, the astronaut, she was part of the team. She helped in carrying out this particular experiment on the International Space Station. The main objective of this experiment was to understand the host immune response of fruit flies in space. So they kind of use this modified European Space Agency Type 1 container. We call it as fly cassettes, which are very tiny fly cassettes. You can see that, and there's a blue color tray that we call it. That is nothing but basically food tray. The flies will feed on that. And the reason we put the blue color is just to ensure that whether these flies are eating the food, because you can easily see through, especially during the larval stage, you can see through whether this blue color is ingested by the fruit fly larvae or not. So just to ensure that they are feeding. And some of the flies will be in this container, and of course it has a lot of ventilation so that they can survive in the space flight, right? So what they did is basically such bunch of fly cassettes with the flies were sent to the International Space Station, and after 12 days they received back. And as soon as they received back, they did the microarray analysis, gene expression studies. What they found is very interesting actually. You can see in this picture here, a microarray data that clearly shows that the genes which are responsible for defense or immune response, especially to the bacterial defense response genes, were significantly downregulated in space-returned samples compared to the ground control, right? More specifically, the antimicrobial peptides such as dosomycin, mechanocovin, atacin C, these are all significantly downregulated. They play a very critical role in innate immune response of the flies in flight samples compared to ground control. So another piece of data I would like to show here is the cells which are actively involved in the phagocytosis process, right? For example, like humans, macrophages, Drosophila also has something called plasmacytocytes. These plasmacytocytes are part of the innate immune response. They are very, very critical for engulfing these microbes which are come in contact with the host cells, right? So that's how they will defend against the microbial invasion. So these cells have that process, we call it as phagocytosis. The cells which are having a phagocytosis ability has significantly downregulated or reduced in case of flight samples compared to ground control. So this is the other picture I would like to highlight. It's a very interesting fact here. This is done by one of our collaborator from UC Davis, Dr. Kimberly and et al. UC Davis group. So this particular fly, I think, born and brought up in the International Space Station, and as soon as they receive these flies on the ground, what they have done is they basically injected very small amount of Bavaria bassiana. It's a entomopathogen fungus. So you can see that within few days it spread all over its body. You can see the white patch everywhere. That clearly indicates the flies came back from the International Space Station, were highly compromised in immune response. They were not able to fight against this fungal infection. So overall, if you look at this experiment, which they have carried out in 2006, that clearly shows that at genetic level, at cellular level, at phenotypic level, they were highly compromised in terms of immune response in the spaceflight environment compared to the ground control. Again, this has been published in 2011 PLOS One paper and 2014 PLOS One paper. You can go through more detailed information on these topics. So the next question is obviously how relevant this information we got in the fruit flies in terms of humans, right, or astronauts in specifically. So I found this particular paper very interesting. In 2004, they have published in Brain Behavior and Immunity, which clearly shows that changes in the neurophil functions in astronauts. I would like to grab your attention to the C panel here. In that figure, which clearly shows, again, the phagocytosis process, the cells which are responsible for phagocytosizing has been significantly downregulated in terms of astronauts' health, astronauts' blood cells actually, right? These are the blood cells or the macrophages of the astronauts which have an ability to phagocytosis were significantly compromised. This is exactly what we have seen in the Drosophila melanogaster as well, right? So that is how the relevance is using the fruit fly model. And another piece of data which again supports, which is published in Journal of Applied Physiology in 2019 more recently, which clearly shows that NK cells, nothing but natural killer cells, these natural killer cells are component of the innate immune response, right? So they were also significantly reduced or impaired in astronauts, which clearly shows that these two publications, and we have many more such publications, which clearly shows that there is a lot of commonality in terms of innate immune response between astronauts and the tiny fruit flies we're using for our spaceflight experiments. So next mission we did was the heart flies, which was on the CRS SpaceX 3 mission. The aim of the study was to understand the impact of spaceflight on the heart structure and function, right? So before I get into the details of methodology and what we found from that information from the data, in fact, this particular experiment was carried out by our one more collaborator from Sanford Burnham Institute, Dr. Bodmer and et al. And Shimla was co-PI in this project actually. So I think it's a very it's important to acknowledge these people before we even talk about these kind of studies, right? Before I talk about the methodology and the results, what they found from this heart flies experiment, it's important to know how relevant heart flies are, the heart of the flies when compared to the human heart, right? It's very important to know that, how relevant it is. Of course, the fly heart does not have the well-defined four-chambered like humans, but it has like a very simple tubular peristaltic pump. And it's also an open circulatory system in the sense, as it pumps, the hemolymph will flood the entire tissue of the Drosophila melanogaster or fruit flies, whereas we have veins. Through that, it's kind of closed system we have. Through veins, the blood will supply to all the organs of the tissue, right? Whereas in case of fruit flies, since it is open circulatory system, it simply floods every tissue by pumping this one. However, if you look at the genetic and molecular mechanisms involved in the cardiogenesis as well as cardiac function, there is a significant overlap between the Drosophila melanogaster and mammals, and in fact humans. And I'd like to point out here two things actually. One is the potassium ion channels, for example, KCNQ and HRG, which are very, very important for cardiac repolarization process, are highly conserved between adult human and the flies. However, they are conspicuously absent in case of rodent model. This makes all the more important why we need to use this Drosophila melanogaster or fruit flies for understanding some aspects of cardiac function or cardiac structure and all that. So that's how it is relevant. This model is. And another interesting fact I need to highlight how relevant the cardiac function in humans as well as in the fruit flies is. There was one interesting article by Wolf et al. In 1991 that clearly shows that the age-related arrhythmias, like the impaired rhythmic movement of heart, like diastolic and systolic function, right? As we age especially from age 50 to 90, up to you go. So there is a significant increase in the arrhythmias or arrhythmic movement of the heart. That is very prevalent in the aging population. You can see it is also very prevalent in everywhere throughout the globe. As we age, there is definitely arrhythmic movement of heart will happen. Similar phenotype is also observed in case of Drosophila melanogaster also. You can see that it's an amazing comparison, which is again made by the Dr. Bodmer et al. The courtesy goes to them. How important this model in order to understand the heart function. For example, I can take this example is one. The first 1 week fly, which is equivalent to 10-year-old boy or a girl, you can see how consistently or rhythmically this is beating, right? The heart pump. It is very consistent, rhythmic, systematically doing because it's very young. Again, if you compare this one with the 7-week-old flies, which is around 70-year-old gentleman or a lady, you can see how inconsistently it is, beating that action. This clearly shows, like humans, in fruit flies as well, as they age old, there is an impaired arrhythmic movement of heart happens. That's an interesting fact. So having this relevance in terms of cardiac function, so Dr. Bodmer et al. Group, what they have done is, with Chemla Bhattacharya's collaboration, they sent this very simple experiment again. Again, you can see this we call it as a vented fly box, where you have a lot of vents so that the flies can breathe a proper oxygen. So what they have done is basically they send these such boxes, few boxes, to the International Space Station. You can also see the vials. These vials will have flies, 15 to 20 flies depending on your experimental plan. We send such 15 vials in each one of those boxes. Such couple of boxes will be sent to the International Space Station. They will spend there for 20-25 days and then come back. So as soon as they came back in the Bodmer lab, what they have done is basically they dissected these hard flies. The flies which exposed to the spaceflight environment were dissected under the microscope and recorded the live imaging of those heart beating actually. So again, I'm not going to go in detail about these results section because still they are not yet published, and there's a lot of information need to be kept secret here actually because we don't have that luxury to reveal as of now. So what I know is from some information regarding this project is basically the flies which came back from the space flight were also having similar arrhythmic movement in the heart, and also there was some changes in the cardiac muscle structure in terms of gene expression studies. So the next question is again, how relevant this information or this study we have Dr. Bodmer et al. Done in the heart flies compared to the astronauts, right? Again, I found very interesting report by the Johnson Space Center, NASA, which is an evidence report which is publicly released on June 12, 2017. You can see that. Again, you can see in the astronauts, they found that the episode of ventricular tachycardia, nothing but basically the impaired cardiac function, similar to what we have seen in the Drosophila melanogaster. In the astronauts as well, you can see such kind of arrhythmic movement of heart when they exposed to the spaceflight environment condition. So another scientific report publications which clearly indicates the Apollo astronauts, the astronauts which went to the moon, they were very highly vulnerable in terms of cardiovascular associated mortality. You can see in the blue color bar here, there are significantly higher cases of cardiovascular associated mortality in the astronauts who went to the Apollo upwell mission compared to the non-flight, low Earth orbit, all flight. So that clearly shows that cardiac function is really one of the big issues we need to be understanding, and model organisms such as Drosophila having so much relevance in terms of cardiac function and gene expression studies is a very good model to understand the cardiovascular issues in space station or in space general. The third experiment kind of we did is, maybe the fourth one, I guess. So this is a very interesting study we carried out when we were in NASA Ames, right? So this is, we call it as Apes Fruit Fly experiment, which is again on SpaceX-4, CRS mission SpaceX-4. So it's an interesting experiment because of the fact that this entire experiment is conceptualized, designed, developed, and executed by the undergraduate students and a couple of master level students. So of course we were there to help them, developing this payload. But basically they are the one who try to do this payload, entire payload. The aim of this experiment was we wanted to understand if there is any correlation between the physiological stress and the behavior of the fruit flies. The reason why we wanted to do that, we know that astronauts confined to the isolated International Space Station or spacecraft will have a significant increase in the physiological stress, and that will impact their activity and behavior on space station. Again, there is a lot of literature available on that, and we wanted to see if there is any correlation between that. So these undergraduate students, what they have done is they developed this hardware called, we call it as FX Habitat, Ames Fruit Fly Experiment Habitat, which is nothing but basically you have like 100 by 100 by 150 mm box. Within that box, you have another small box that is around 40 by 95 by 50 mm in size. And in this small box, we have two chambers because we wanted to send two different types of flies, right? So one is wild type, and again you can see that blue color food. That is, the food which flies feed on. So basically, we wanted to send two types of fruit flies here. One is wild type. Wild type is nothing but, when you throw this banana in your corner of home, you will get these buzzing flies, right? Those are wild type flies, which are not very genetically homogeneous in terms, right? And we wanted to send another mutant called a stress-resistant mutant called Indy. Indy stands for I'm not dead yet. These flies are really, really long-lived, like 20-25% more longer than the, they live longer than the wild-type flies, and also they are resistant to the various kinds of stresses, like physiological stress, the heat stress and all that. So that is another model, another advantage of using fruit flies, because they are genetically very amenable in the sense these are commercially available flies, various kinds of mutants and overexpressed transgenic flies are available commercially. You can just order them, get them from the Bloomington Stock Center, or in India there are a couple of institutes like Mysore University of Mysore is also having that kind of facility where you have the stock of these mutants and transgenics. You can easily get them and study the individual gene function by using such kind of mutants and transgenic plants. That is another advantage of fruit flies, which is kind of very difficult in the higher animal model. So we wanted to compare between the wild type and the Indy resistant mutant because we wanted to see if how vulnerable or how resistant in spaceflight condition in terms of the physiological stress and the changes in the behavior. And of course, the entire box is having this video monitoring system so that you can, you can track the real time the behavior of these flies from the space station, and it has vents and fans so as to circulate the good air circulation inside the habitat. Then UV and IR light sources are very important because these flies, like humans, they also have a circadian rhythms, like they can also biological clocks. So they need to understand light and dark cycle. Then only it will not impact the behavior. Otherwise, the light and dark cycle will have impact on the behavior. We wanted to make sure that it does not affect that. And of course, there are other sensors so as to maintain the temperature, relative humidity, carbon dioxide, oxygen level intact or optimum level inside the habitat so that it will not interfere with the fly behavior. So again, very simple experiments we did here. Such boxes we sent to the International Space Station, bring them back as soon as we receive them flies. So in the lab, this we in-house developed a MATLAB program. Through that, we track the behavior of those flies in collaboration with the Stanford University. Then what we found is basically these flies came back from the International Space Station where it's highly active. In fact, they were like hyperactive in space compared to ground control flies in terms of locomotor behavior as well as in terms of flying behavior, right? And also sleep behavior is affected significantly. Again, I would like to go through this Mal et al. Paper, which is which is important. Um, they clearly shows that there is a significant impact on the circadian rhythm as well as sleep behavior is impacted in the space station, in the Chinese space station actually, right? So that experiment is also cool. So the reason why sleep behavior is affected, yet to be understood, the cellular mechanism and mechanistic role of the sleep behavior is changes need to be understood very thoroughly. But however, this is exactly what we have seen in the astronauts as well, actually. In fact, the astronaut sleep behavior is erratic on the International Space Station. And sometimes they will take this melatonin or some other pharmaceutical intervention so as to have a good night's sleep on the International Space Station. Again, it's very relevant to the, what the sleep behavior or hyperactivity have been observed in case of fruit flies, right? But one more interesting fact came out from this study is if we have interventions such as nutritional intervention or pharmaceutical intervention, you can actually reduce the physiological stress level, and that will have a normalcy in the behavior. And that's why you can see the astronauts will have a very highly nutritious food, and also, if necessary, there is pharmaceutical intervention so as to have balanced physiological stress level so that it will not impact the activity they're carrying out routinely on the International Space Station. So with that three or four experiment, Dr. Shimla Bhattacharya and group is kind of convinced NASA to have a dedicated research program on fruit flies so that we can follow up some of the studies we carried out and also look at the astronauts' health more molecular mechanistic point of view. So NASA was very generous and they initiated this program called Fruit Fly Lab series of experiment, which is a dedicated program for fruit fly lab experiment, only for the fruit flies studies on the International Space Station. And first installment of that was Fruit Fly Lab 1. I was part of this mission again, which was flown on SpaceX 5. This was a tech demo validation mission actually, because we built the new hardware. We wanted to test this hardware, whether it works on the International Space Station or not. So along with that, we also had science objectives. Again, this is on the innate immune response. But the difference between this experiment and very first experiment that Shimla Bhattacharya's group carried out in 2006 was, in that experiment, in the FIT experiment, what they have done is basically they sent the fruit flies to the International Space Station. Then they got back within 12 days. Then they injected the E. coli on them and looked at the immune response from the host point of view. In this case, what we have done is we sent both fruit flies as well as another microbe, the human pathogen called Serratia marcescens, to the International Space Station. There, we wanted to study host pathogen interaction. Unfortunately, because of some technical issues, we could not get the live flies back, but we got these Serratia marcescens, which are exposed to the space station for 30 days, came back from the space to the Earth. As soon as we received those microbial sample, what we have done is we injected these marcescens, which are exposed to the space flight, to the ground flights, and looked at from the point of pathogenicity. So the difference between those, their host point of view here and the pathogenicity point of view. Okay, of course, there's a lot of information available on the microbial virulence and pathogenicity goes up in the space as well as in simulated microgravity. And we wanted to test that hypothesis in fruit flies as well. So in fact, we recently published this research in the Nature Microgravity. You can go through that. I mean, just a couple of months back, we published this paper, which clearly shows that the Serratia marcescens human pathogen, which is exposed to the spaceflight as well as simulated microgravity condition, showed a significant enhancement in the death of the flies, indicating their increased virulence as well as pathogenicity with the same microbial load. So, of course, we also had a few larvae, intact larvae from the same spaceflight mission. We did some proteomic studies as well. I'm not going to go into the details of all these proteins. What I'd like to highlight here is there are some proteins which are specifically associated with muscle cuticle chitin biosynthesis were significantly decreased in spaceflight larvae compared to the ground control. Again, how relevant this one? The exoskeleton of the insects are equivalent to the human bone and muscular structure, right? So again, we know that there is a lot of changes in the bone as well as muscle structure and muscle function. Similar kind of the proteins were associated with in flies as well, changed in fruit flies also. So then by that time, I had to come back to India because of some reason. So then, Dr. Shamrocks Group, Bhattacharya's group, followed up on SpaceX 9 and SpaceX 11, FFL2, FFL3, and we hope that, we're going to get a lot of good information from these flight missions they have done, and hopefully we'll get the publication soon out of them. We'll get to know the information on this. So once I come back from the US, I was always interested to initiate such kind of work in India, because at that time nobody was doing. I mean, sporadic experiments were carried out not on the flies, but using the simulated microgravity and hypergravity work. In such example is Dr. Pandit Vidyasagar's lab at Pune University. Very sporadic labs were working on this kind of work, but I was very keen on initiating this space flight experiments. And luckily and coincidentally, there was an announcement by the government of India, Gaganyaan program, and ISRO immediately called for this announcement of opportunities, and we grabbed this opportunity in collaboration with Dr. Srija Lakshmi, Indian Institute of Space Science and Technology, and we proposed this study, and ISRO recommended this study, and hopefully we'll have, if everything goes well, the first fruit fly mission will go from the Indian soil in the Gaganyaan program in the next few months or maybe a couple of years. So, the objective of this study was we wanted to study the kidney stone formation in fruit flies. The reason, again, it's very relevant to the astronauts' health, because we know that astronauts staying on the International Space Station are very vulnerable for the kidney stone. Because so with that, I kind of convinced in the previous slide that the stuff of the model is very, very important for understanding not only human biology, specifically the astronauts' health point of view, right? So once I come back from the US in 2008, I was always very keen to understand what kind of studies need to be done and initiate such kind of work in India. And of course, at that time, not many people were working on this kind of studies, not only in India, but also in abroad. So I was very keen on initiating this, space flight studies using the fruit fly model for human health understanding, right? So coincidentally and luckily, there was an announcement by the government of India. They initiated this program called Gaganyaan, and ISRO immediately announced an opportunity for sending the biological payloads to the Gaganyaan. We grabbed this opportunity and proposed study in collaboration with Dr. Srija Lakshmi, an IIST scientist, Trivandrum, and so with that collaboration, we studied this. We proposed this study, which is mainly deals with the kidney stone formation in fruit flies. Again, it is a very relevant model, right? Kidney stone formation, because you know that astronauts who are there on the International Space Station will always have a very high vulnerability in terms of kidney stone formation because of the leaching of the bone minerals as well as calcium that will go and deposit into the kidney and forms a calcium oxalate, and that will lead to the kidney stone formation, right? So similarly, in case of fruit flies also, fruit fly malphizian tubules are equivalent to human kidneys. You can see this beautiful picture, a scanning electron microscope, that clearly shows that the kidney stones, the calcium oxalate crystals, were deposited in the malphizian tubules, exactly similar to the human's kidney stones in humans, right? So we wanted to understand how kidney stone formation happens both in space as well as in case of Earth condition. And this, hopefully, the fingers crossed, and hopefully we'll get to fly this opportunity when the Gaganyaan program starts, right? So this has been recommended by the ISRO, and we're very looking forward to fly this mission from the very first from the Indian soil. So with that, I try to explain how relevant this model for understanding the human biology in general and astronauts' health in specific, right? However, it's always difficult to extrapolate to the humans, right? So that's why you need to have the studies they have done, like NASA's twin study, right? Where multidimensional analysis of year-long human spaceflight program has been done. In this case, what they have done is basically Scott Kelly and Mark Kelly. They are genetically identical twins. They were there. They participated in this research program. And Scott Kelly, being 6 hours younger than the Mark Kelly, was up in the International Space Station for 340 days, whereas Mark Kelly was on the ground being controlled to his sibling. So what they found is they have done very, the comprehensive study of using biochemical, cognition, epigenomics, gene expression, immune response, metabolomics, probiome, microbiome, and proteome, various kinds of studies. And interesting facts come out from that study is actually very encouraging. In the sense, for example, they found that there's an increase in the telomere length, right, which is directly relevant to the aging and there is changes in the DNA methylation patterning, there is changes in the immune cell response, cardiovascular, cognitive effects. But important thing is, once they came back to the Earth, everything came to the normalcy. So that is very encouraging result, right? Which will give a lot of confidence to the to the to the research community to explore further space. So with that, I still remember in one of our conversation with the NASA's the management community at NASA Ames Research Center, what they have said is, if we hadn't done 30 to 40 years of research, fundamental biology, space biology research, using these tiny model organisms such as microbes, nematodes, fruit flies, and mice, probably we wouldn't have a confidence today to send these fruit flies to the we wouldn't have a confidence to send Scott Kelly to the ISS for 340 days, which kind of sums up how important these tiny model organisms, including fruit flies, to understand human health in general and astronauts' health in specific. With that, I would like to thank all my lab mates. We have been tremendously helpful in terms of carrying out some of these experiments on the ISS when we were working at NASA Ames Research Center between 2011 to 2018. And you can see Shamla Bhattacharya sitting there. She's been real mentor to me, and she has been inspirational for me to carry out such kind of work at NASA and even in India. In fact, when we proposed this Gaganyaan mission, I was constantly in touch with her, and she was being very supportive of initiating this kind of work in India. And I hope she will continue to work in this area of research and inspire young people like us to take this space biology forward. In various developing nations like India and other space agencies, right? So, in fact, I would highly recommend you guys to take a look at Sharmila's TED Talk, which is being uploaded on the web YouTube very recently, and she's been introduced none other than Shah Rukh Khan. So she's real celebrity in that sense, and she has given real good picture of how space biology and fruit fly research is important for human exploration point of view. With that, I'd like to conclude my again. Thank you, Dr. Bala, as well as Dr. Shija Lakshmi, for giving me this opportunity to carry out or kind of talk about my research work, which I've done at NASA Ames. Thank you. I'm happy to take a few questions.
Okay, thank you, Dr. Ravikumar, and thanks for accepting the invitation to speak on this webinar.
Sure.
Webinar. Of course, it was very new for us about the flies in microgravity and to our participants as well. Right. Here we have questions from Manan Desai and Debadatta. So I'll read one by one. Can artificial gravity help alleviate adverse effects on astronauts' health?
Artificial gravity? So in the sense hypergravity. They're talking about hypergravity. Yeah. So there are not very convincing research has been done in that, but there are some sporadic literature which is available on the net. You can easily go through. In fact, in our own experiments, we have done that when we expose these flies to the hypergravity environment, for example, 3G, 3 times the gravitational force of Earth, right? That will give actually the immune boost. So in that sense, it's helpful. It can mitigate some of the impact created by the microgravity environment. That is, again, in the model organisms, people have been tested out. In model organisms, hypergravity have been utilized, artificial gravity has been utilized to mitigate the impact of microgravity environment on the model organisms. I don't know much literature on the astronauts themselves, but astronauts, before astronauts will be launched to the space station, they will be exposed to this gravitational force, just to make them harder, because that when there is a launch as well as a re-entry into the atmosphere, there is a hypergravity experience they will experience. So in order to mitigate that, astronauts will be exposed to the kind of hypergravity environment to cope up this the descent and landing as well as the launch. But again, as you said, artificial gravity is possibility. There is a possible chance that it can mitigate some of the effects which has been established in model organisms like fruit flies and mice. But in case of astronauts, I don't see any experiments carried out that kind of studies.
Okay, thank you. There's another question. What are the advantages and disadvantages of using microorganisms as model organisms in spaceflight? From Devadatta.
Okay, so microorganisms. He's talking about microorganisms.
Yeah, you have to pin his video. Yes. So the question is, what are the advantages and disadvantages of using microorganisms as model organisms in spaceflight?
Well, so advantages is, especially people have been doing a lot of microorganisms. For example, especially they're looking at the human pathogens like Salmonella, Pseudomonas, Riccium arsensis. If you can look at, I would highly recommend you to take a look at the Dr. Cheryl Nicholson paper. She's been pioneer in doing such kind of work, and also the Dr. Kasturi Rangan. Venkateshan, Dr. Kasturi Venkateshan, actually, so from JPL. So he has done a lot of this work. Advantages is obviously they are looking at in terms of these are small organisms, they are obviously having a lot of advantage in terms of space and all that. But also they are human pathogens. You can directly get the answers, like whether they are virulent, how they are behaving in space station in terms of virulence, in terms of pathogenicity, and how it is exactly whether they will similar way they are behaving in ground control as well. So those are some of the advantages we have. And of course, disadvantages is the in terms of molecular level, mechanistic level. These again, as I said, there is a lot of genetic commonality between humans and the microorganisms, as well as many of these fruit flies and nematodes and mice, right? So that commonality will give you the understanding of molecular mechanistic of particular disease or particular hypothesis, right? So that is an advantage. Disadvantage, of course, you cannot. Sometimes it is very difficult to extrapolate to the human biology. For the obvious reasons, that, right? It is difficult to. That's why we need to have the importance of having different model organisms so that you can confidently extrapolate that result to the human conditions. But another disadvantage, a lot of time you have to be very careful in carrying out these microorganism studies on the International Space Station, because they are more worried about mutations that can increase the virulence and which may impact the astronauts' health in space. When you're going to do the experiment, that could end up infecting the astronauts or contaminating the whole environment within the space station, right? So that is kind of containment needs to be taken care of. So these are some of the advantages and disadvantages of using the the microorganisms.
Thank you. And the next question is from Anand Desai. What was the reason for suppressed immune response and downregulation of the genes as shown in the microarray data?
Okay, so the reason why it is okay. So this is again, the many of the studies. This is another disadvantage of carrying out many of the flight experiments, right? Because it is very difficult to follow up. Because in fact, at NASA only, that's why they have this dedicated program they started. But until now, these are kind of observation studies, right? Why particular set of genes have downregulated is very difficult to understand, because it is the follow-up studies need to be done, and we haven't done yet. Okay, so that's why we don't know what is that it is making these particularly immune compromised response or genes which are specifically responsible for bacterial defense is downregulated in the space station. So that kind of studies need to be done. For overall, it is kind of, and I think in the space station, the microgravity, they sense it as a stress, physiological stress, and that physiological stress is very general term again. So to answer a specific question, we don't know yet why this specific set of genes have downregulated. But my understanding is we need to do more kind of research so that we can easily understand why that kind of downregulation happening in case of specific set of genes in space station. Again, spaceflight experiment being very expensive, it is very difficult to follow more and more in studies. So I think it requires more information, more research and more information to understand the specific mechanism, why there is a change in the cardiac function, why there is a change in the gene expression of immune response, why there is a change in the muscle and the chitin biosynthesis pathways. Okay, all that needs to be studied.
Okay. Okay. From Devadatta Das, something slightly similar to that. Why does muscle and bone degradation occur in space environment?
Okay, again, so because I think it is use and disuse theory, right? So generally you use more muscles and bones that to tend to be healthy. That's why you do the exercise and all that. In microgravity, what happens? Basically, these are become useless. Although you have legs and everything, because you're floating, you are even under microgravity, and there is no mechanical loading on these muscles and bones, right? As a result of that, you will start losing the integrity and structure and function of those bones and muscles. Actually, it's basically the lack of mechanical loading on these bone and muscle that will have an impact on the loss of bone as well as loss of muscle structures. Absence of mechanical loading. One sentence answer for you.
Okay.
Yeah.
And similar line, Navanil has another question. How is kidney function affected in microgravity? Is there any increase on the kidneys?
Yes. So again, there are a lot of literature available. You can go through that. So basically, what will happen is you have these, because of the microgravity, the muscles as well as bones will leach out a lot of these calcium and other minerals that will go and deposit in the kidney. Okay, that will form a kidney, calcium oxalate, and as a result of that, that will form a kind of kidney stone formation, right? So kidney stone formation is one of the major issues in the astronauts. That's why they take this pharmaceutical intervention like potassium citrate. Potassium citrate is one of the, which is commonly given to the kidney stone problems in Earth as well. So they will give this potassium citrate to reduce the kidney stone formation. It is basically because of the leaching of the bone mineral and bone calcium in the microgravity. That is because of the loss of or lack of mechanical loading, as I said in the previous question, right? So because of that, there is a leaching of some of the minerals that will go and deposit in the kidney and become a kidney stones.
Okay. One more question from Navanil. How is blood clotting affected in microgravity?
What is that? Sorry?
How is blood clotting affected in microgravity?
Oh, okay. How is blood clotting affected? So, to frankly tell you about that, I don't have that. I don't have information on that because we have not done blood clotting kind of studies in fruit flies. But what we have seen is there is some changes in the drosophila blood cell number in that fit experiment which we have carried out, right? So we specifically looked at the numbers of, Shamrocks Bhattacharya's group specifically looked at the blood numbers, but I don't know the exact mechanism how blood clotting is impacted in microgravity because that kind of experiment we have not done in the ISS.
Okay, right. So that is carried out so far, blood clotting experiment?
No, actually, so we just looked into the numbers of cells, whether the blood cell number is going up or decreased in the space station experiment. Other than that, we have not done because we were having very less samples from that experiment. So we had to do many other experiments on the immune response, right? So this was just part of that experiment. But yeah, we couldn't do that.
Okay, so in line with this question, I'm just curious to know. Suppose an astronaut is trying to do some activity.
Okay.
Maybe a small knife has cut the skin.
Yes.
So now the blood starts coming out.
Right.
And how soon does it stop? Have they tried this?
Wound healing, basically the wound healing, right?
Yeah.
Yeah, it's kind of wound healing experiment. I'm sure they might have done some experiments in the using rodent models or some rodent models or mice or rat model, but I'm not sure about in any other model organisms they have tried this actually, right? Because I know for the fact that in fact on the International Space Station they will also dissect the mice because they have a capability to dissect the mice and separate out their brain, their all the muscles, bone, and everything. So dissection set is there. So I don't know how exactly this will play in terms of wound healing mechanism on the ISS. In terms of flies, we have not done such kind of experiment.
Okay. All right. There's one question from Hari Krishnan. Okay. Will the muscles go vestigial if we spend significant amount of time in space flight?
Yeah, that's what the worry is, right? If you look at the report of the NASA, it is very alarming. Like 1% if you're staying on the ISS, which is again in the protective layer of the biosphere, right? So you are going to lose 1 to 2% of bone mass and 20-25% of muscle loss, which is huge. And if you continue doing long-term experiments in long-term space exploration like Mars or wherever, go to the other planets, which would take around, for example, Mars would take around 500 round trip, right? Roughly. So including the stay for a few days there. So this is a very significant impact. We don't know whether they will go vestigial, but definitely it's going to be dysfunctional. They are going to be irreparable chances when they go beyond the, if you don't take the pharmaceutical intervention or exercise. And that's why it is mandatory to have these astronauts exercise, physical exercise every day, 2 to 2.5 hours on the ISS, so that they can keep this one health very intact. But yeah, I don't know whether it will go to the vestigial, definitely, but there is a significant loss of functions, and it may be irreparable if you go beyond that and if you don't take any intervention.
Okay. Okay. So I think we are right on time.
Yeah.
Ready? If there is any other question. Okay, we are already 10 minutes past our schedule. Okay. So maybe we can stop here. Yeah. Okay. Sure. So thank you, Dr. Ravikumar.
Thank you. Thank you. It's nice knowing you. Thank you, Dr. Mala. It's nice knowing you, and we'll be in touch with you.